Dear viewers, this video is part 2 of the series on lipid disorders under the Prescribon Medical Education series. Free fatty acids are transported in the blood in combination with albumin. When fat stored in the adipose tissue is to be used elsewhere in the body to provide energy, it is first transported from the adipose tissue to the other tissue. It is transported mainly in the form of free fatty acids. This transport is achieved by hydrolysis of the triglycerides back into fatty acids and glycerol. Upon leaving fat cells, fatty acids combine with albumin molecules in the plasma proteins. Fatty acids bound in this manner are called free fatty acids or non-esterified fatty acids. The concentration of free fatty acids in the plasma is 15 mg per deciliter, which is a total of only 0.45 gram of fatty acids in the entire circulatory system. Even this small amount accounts for almost all of the transport of fatty acids from one part of the body to another for the following reasons. 1. Despite the minute amount of free fatty acid in the blood, its rate of turnover is extremely rapid. Half the plasma fatty acid is replaced by new fatty acid every 2 to 3 minutes. Almost all the normal energy requirements of the body can be provided by the oxidation of transported free fatty acids without using any carbohydrates or proteins for energy. 2. Conditions that increase the rate of utilization of fat for cellular energy also increase the free fatty acid concentration in the blood. In fact, the concentration sometimes increases 5-fold to 8-fold. Such a large increase occurs especially in cases of starvation and in diabetes mellitus. In both these conditions, the person derives little or no metabolic energy from carbohydrates. Dear listeners, by the way, this educational video is made possible by Prescribon, the makers of e-prescription software for digital prescriptions available freely for doctors on the website www.prescribon.com. Doctors who are interested in this e-prescription can freely avail of its vast collection of more than 100,000 e-prescriptions. In the post-absorptive state, after all the chylomicrons have been removed from the blood, more than 95% of all the lipids in the plasma are in the form of lipoprotein. These lipids are small particles, much smaller than chylomicrons, but qualitatively similar in composition, containing triglycerides, cholesterol, phospholipids, and protein. The total concentration of lipoproteins in the plasma averages about 700 mg per 100 ml of plasma, that is 700 mg per deciliter, and can be broken down into the following individual lipoprotein constituents. Blood concentrations of cholesterol in milligram per deciliter 180, phospholipids 160, triglycerides 160, protein 200 milligrams. Types of lipoproteins. The amount of lipid in a lipoprotein determines its density because lipids are not as dense as water. The lipoproteins' density and size, content of triglycerides, cholesterol, phospholipids, and protein follow in order. Nanometer, 1 nanometer means 1,000th of a micrometer. Chylomicrons less than 0.95, more than 75 nanometer in size, triglyceride 80 to 95%, cholesterol 3 to 7, phospholipid 3 to 6, and protein 1 to 2. Aside from the chylomicrons, which are very large lipoproteins, there are 4 major types of lipoproteins. Very low density lipoproteins, VLDLs, which contain high concentrations of triglycerides and moderate concentrations of both cholesterol and phospholipids. VLDLs density 1.006, size 30 to 75 nanometer, triglycerides 59, cholesterol 15, phospholipid 5, and protein 10. Intermediate density lipoproteins, IDLs, which are VLDLs from which a share of the triglycerides has been removed, so the concentrations of cholesterol and phospholipids are increased. 3. Low density lipoproteins, LDLs, derived from IDLs by the removal of almost all the triglycerides, leaving an essentially high concentration of cholesterol and a moderately high concentration of phospholipids. That is low density LDL, 1.04 density, 18 to 25 nanometers, triglyceride 10, cholesterol 45, phospholipid 22, and protein 20 to 25%. High density lipoproteins HDL contain a high concentration of protein, about 50%, but much smaller concentrations of cholesterol and phospholipids. HDL density 1.1, size 4.5 to 12, triglycerides 3, cholesterol 2, phospholipids 27, and protein 48. Formation and function of lipoproteins. Almost all the lipoproteins are formed in the liver, which is also where most of the plasma cholesterol, phospholipids, and triglycerides are synthesized. In addition, small quantities of high density lipoproteins are synthesized in the intestinal epithelium during absorption of fatty acids from the intestines. The primary function of the lipoproteins is to transport their lipid components in the blood. The VLDLs transport triglycerides synthesized in the liver mainly to the adipose tissue. The other lipoproteins are especially important in different stages of phospholipid and cholesterol transport from the liver to the peripheral tissues or from the periphery back to the liver. There are special problems of cholesterol transport in relation to the disease atherosclerosis, which is associated with the development of fatty lesions on the insides of arterial walls, discussed later. Fat deposits. Large quantities of fat are stored in two major tissues of the body, the adipose tissue and the liver. The adipose tissue is usually called fat deposits or simply tissue fat. Adipose tissue. A major function of adipose tissue is storage of triglycerides until they are needed to provide energy elsewhere in the body. Additional functions are to provide heat insulation for the body and secretion of hormones such as leptin and adiponectin, which affect multiple body functions, including appetite and energy expenditure. Fat cells, that is adipocytes, store triglycerides. The fat cells, adipocytes, of adipose tissue are modified fibroblasts that store almost pure triglycerides in quantities as great as 80% to 95% of the entire cell volume. Triglycerides inside the fat cells are generally in a liquid form. Fat cells can synthesize very small amounts of fatty acids and triglycerides from carbohydrates. This function supplements the synthesis of fat in the liver. Tissue lipases permit exchange of fat between adipose tissue and the blood. Large quantities of lipases are present in adipose tissue. Some of these enzymes help deposit triglycerides from the chylomicrons and lipoproteins. Others cause splitting of the triglycerides of the fat cells to release free fatty acids. Because of the rapid exchange of fatty acids, the triglycerides in fat cells are renewed about once every 2 to 3 weeks, which means that the fat stored in the tissues today is not the same fat as that was stored last month, thus emphasizing the dynamic state of storage fat. Liver lipids. The principal functions of the liver in lipid metabolism are to 1. Degrade fatty acids into small compounds that can be used for energy. Synthesize triglycerides mainly from carbohydrates but to a lesser extent from proteins as well. And 3. Synthesize other lipids, especially cholesterol and phospholipids, from fatty acids. The liver cells, in addition to containing triglycerides, contain large quantities of phospholipids and cholesterol, which are continually synthesized by the liver. Also, the liver cells are much more capable of desaturating fatty acids than are other tissues, and thus liver triglycerides normally are much more unsaturated than the triglycerides of adipose tissue. This capability of the liver to desaturate fatty acids is functionally important to all tissues of the body because many structural elements of all cells contain reasonable quantities of unsaturated fats and their principal source is the liver. Use of triglycerides for energy. Formation of adenosine triphosphate ATP. The dietary intake of fat varies considerably in persons of different cultures, averaging as little as 10 to 15% of calorie intake in some Asian populations to as much as 35 to 50% of the calories in many Western populations. For many persons, the use of fats for energy is therefore as important as the use of carbohydrates. In addition, many of the carbohydrates ingested with each meal are converted into triglycerides, stored and used later in the form of fatty acids released from triglycerides for energy. Hydrolysis of triglycerides into fatty acids and glycerol. The first stage in using triglycerides for energy is their hydrolysis into fatty acids and glycerol. Then, both the fatty acids and the glycerol are transported in the blood to the active tissues where they will be oxidized to give energy. Almost all cells, with some exceptions such as brain tissue and red blood cells, can use fatty acids for energy. Glycerol, upon entering the active tissue, is immediately changed into glycerol 3-phosphate and enters the pathway for glucose breakdown and is used for energy. Before the fatty acids can be used for energy, they must be processed further in the mitochondria. Large amounts of adenosine triphosphate, that is ATP, that is packets of energy for cells, are formed by oxidation of fatty acids. Formation of acetoacetic acid in the liver and its transport in the blood. A large share of the initial degradation of fatty acids occurs in the liver, especially when large amounts of lipids are being used for energy. However, the liver uses only a small proportion of the fatty acids for its own intrinsic metabolic processes. Instead, when the fatty acid chains have been split into acetyl coenzyme A, two molecules of acetyl coenzyme A condense to form one molecule of acetoacetic acid, which is then transported in the blood to the other cells throughout the body where it is used for energy. Normally, the acetoacetic acid and beta hydroxybutyric acid that enter the blood are transported so rapidly to the tissues that their combined concentration in the plasma seldom rises above 3 mg per deciliter. Yet, despite the small concentration in the blood, large quantities are actually transported, as is also true for free fatty acid transport. The rapid transport of both these substances results from their solubility in the membranes of the target cells, which allows almost instantaneous diffusion into the cells. Synthesis of triglycerides from carbohydrates. Whenever a greater quantity of carbohydrates enters the body than can be used immediately for energy or can be stored in the form of glycogen, the excess is rapidly converted into triglycerides and stored in this form in the adipose tissue. In humans, most triglyceride synthesis occurs in the liver, but minute quantities are also synthesized in the adipose tissue. The triglycerides formed in the liver are transported mainly in the VLDLs to the adipose tissue where they are stored. Dear viewers, this is the end of part 2 in this 4-part series on lipid disorders. For other videos in this series, see Prescribon Medical Education series, Lipid Disorders.